Most of you have probably heard the term protein. We're used to thinking of proteins as components of our diet, or of course, as making up our muscles that we use to move around and exercise. Now, both of these are very important roles for proteins. But as biochemists, we usually talk about proteins as part of their role within the cell. Proteins are one of the key macromolecules in our cells. They, of course, can play a structural role in helping to hold our cells together, but they also have a wide variety of different functions inside of our cells as well. So our proteins are actually one of the primary agents of our various biological functions. They're involved in things like catalyzing reactions. They're involved in transporting different molecules around our body or within our cells. Again, they do play a structural role in holding our cells together. And they're also involved in motion, right? So that's our typical muscle proteins would be involved in that motion role down here. So they're actually one of the most important biological molecules that we'll look at in this semester. Now, despite the complexity of our proteins, they're actually built up from relatively small, simple molecules that we call amino acids. So our proteins are biopolymers. So polymers, of course, refer to a molecule that's built up by connecting lots of smaller molecules together. And those smaller molecules, those monomers, we call them, are these simple amino acid molecules. So we're going to start by looking at the details of our amino acid structure so we can understand how our individual amino acids behave. And then we'll see how we can link those together to make proteins and then make all the various functions of our proteins within our cells. So our proteins are built up from our alpha amino acids. Our amino acids have several key properties that will allow them to build our proteins and carry out these various functions. Of course, the most critical property is that they can polymerize easily, right? If we're going to be able to build these together to make our proteins, they have to have the chemical functional groups to allow them to link together. They also have useful acid-base properties, so they can participate in some of those very common acid-base reactions that we have in our body. They have various physical properties. So this allows us to create a wide variety of different proteins, right? Our amino acids were only fit in one category in terms of our physical properties, such as hydrophobicity or hydrophilicity, then our proteins would pretty much all look the same. But we actually have quite a wide variety within our amino acids so that we can then have a wide variety in our proteins as well. So that they have various physical properties and then various chemical functionality as well. So our basic structure for our amino acid is actually quite simple. To build an amino acid, we just need an alpha carbon at the center, and then we're going to have 4 different groups attached to this in a, of course, tetrahedral arrangement. Right? Whenever we have 4 things attached to a carbon, they always our bonds always push each other away into that perfect tetrahedral shape. Now, 3 of these things will be the same for almost all of our amino acids. We'll always have 1 hydrogen, 1 carboxylic acid, and 1 amine group. So these are shared between all of our amino acids. And then this last group down here, marked with an R, is our variable side group. So this group will change between different amino acids. So in our body, we commonly have 20 different amino acids, and each of those 20 different amino acids will have something different attached here at this 4th position in order to create the variety within our amino acids. Now, of course, whenever we have 4 unique groups attached to a single atom, in this case our alpha carbon, that creates a molecule that can be chiral, right? You should remember this from your organic chemistry class. So we actually have 2 different chiral forms of our amino acids. In biochemistry, we commonly use an L and a D to distinguish between our 2 different enantiomers here, or 2 different chiral forms. And within our natural proteins, we really only see our L forms of our amino acids. So our body is actually quite chiral overall. We have a high preference for certain chiral molecules or enantiomers over their corresponding enantiomer. So in the case of our amino acids, we always prefer that L form. Now, again, as I mentioned earlier, one of the key properties of our amino acids is that they need to be able to polymerize. So we accomplish this using that acid, carboxylic acid, and amine that are attached on every amino acid. So here's our acid, here's our amine. And of course, you might remember from organic chemistry that an acid and an amine can react together in a condensation reaction. We would specifically refer to this as a nucleophilic acyl substitution reaction. It allows us to link these together and create an amide functional group that now combines our two different amino acids. Now, of course, you might notice here the name for amino acid actually comes from the fact that we have an amine and an acid combined here together in this small molecule, right? So the amine gives us the amino portion of our name, and the acid comes from the carboxylic acid functional group. So we'll look at this more closely later, but this bond here, our amide bond, is going to be critical for linking our amino acids together to form our long protein biopolymers. Now, of course, if we just link the same amino acids together over and over again, we would get a relatively boring protein, right? So the key variability of our amino acids that will then make our proteins highly variable and able to carry out all those various functions, catalyst, transport, motion, etc., requires, of course, that variability in our amino acid side groups. So I want to take a closer look at these amino acid side groups so you can see what kind of variety we have to work with as we build our proteins. So we generally break apart our amino acids into 5 different groups based on the structures of their R groups, their R substituents. We have amino acids with nonpolar aliphatic R groups. And I should be clear that nonpolar is just referring to the R group, not necessarily the amino acid overall. We have aromatic amino acids based on that R group, so that they would have an aromatic group within their side chain. We have more polar, uncharged R groups. We have positively charged R groups, and we have negatively charged R groups. So these are the 5 main categories that we'll typically see in biochemistry. Of course, there's other ways to organize our 20 different amino acids and categorize them based on some of the shared chemical functionality between different amino acids, but this is the most common classification. So I'm going to quickly show you what the different amino acids look like. And of course, we'll spend more time talking over the details of what those look like, what their similarities and differences are as we dive more into amino acid structure later. So first off, here's our nonpolar amino acids. So we have glycine in the top left. This has our smallest side group with just a hydrogen. Then we have alanine, proline, valine, leucine, isoleucine, and methionine. Notice for all of these, the primary components, right, are just carbons and hydrogens because these are all nonpolar hydrophobic amino acids R groups. Notice proline up here is a bit unusual because it actually has its R group connecting to our amine here also, right? So you form this little ring where it bends around and attaches back to our nitrogen. Methionine also is slightly unusual because it has a sulfur and not just our carbons and hydrogens. Our next set of amino acids are the aromatic amino acids. So notice we have a 6-membered carbon benzene ring in each of these. We have phenylalanine, tyrosine, and tryptophan. All of these are classified based on again, that aromatic group, but they do vary somewhat in what other function groups we have, right? We have an alcohol here in our tyrosine that would make it a bit more polar, and we have an amine here in our tryptophan. Next, we have our polar uncharged R groups. So we have serine, threonine, cysteine, asparagine, and glutamine. So these amino acids, of course, are polar and can also form hydrogen bonds, for most of them anyways. Cysteine, of course, is the exception with our thiol group here. It doesn't really form strong hydrogen bonds, but as we'll see, it can form these disulfide bonds that will actually be quite important for protein structure. So all of our polar groups in here, of course, they're what are allowing us to make those hydrogen bonds. And they're, of course, what also make these amino acids relatively polar overall. So we have a variety of alcohols, or of course, our amides down here for asparagine and glutamine. Next, we have our positively charged R groups. So these include lysine, arginine, and histidine. So you'll notice we usually have amines here in our side group to provide our positive charge. Amines are the most common biological base. Histidine here actually is shown in the neutral form. It's this nitrogen that normally gets protonated in order to form our positively charged histidine. Histidine also actually has a little ring structure and is aromatic, but it's normally classified with our positively charged groups because of that positive charge. And finally, our last two common amino acids are negatively charged. So these are aspartate and glutamate, and here we're using our carboxylic acid to provide that negative charge. So again, these amino acids are all classified based on the properties of their side group. However, just because their side group might have a specific property, such as being more hydrophobic, that doesn't mean that the amino acid overall will behave just like the side group, right? You have to consider that we have our carboxylic acid and our amine attached to that alpha carbon for every amino acid, right? So you've got the alpha carbon, and you always have a carboxylic acid and amine attached there, regardless of what side group you might have. And of course, you have that hydrogen as well. So for something like glycine, it actually ends up being quite polar, even though its side group is considered nonpolar. So this scale here is actually showing the relative hydrophobicity of each of our amino acids. So this is actually an experimental result from a paper done in the 1990s. So the primary authors here were White and Wimley. They're the scientists that pioneered this work. So what they did is they were looking at whether the amino acid would prefer to go into a more nonpolar solvent, so this is octanol, versus a more polar solvent of water. So our free energy over here on the y-axis would be negative if it prefers the more nonpolar solvent, or positive if it prefers the more polar solvent. So what you'll notice is that we have basically a set of around, let's see, this is 8 amino acids that tend to prefer more nonpolar, and we have the remaining 12 amino acids that actually tend to prefer more polar environments. So a lot of these that are most nonpolar are actually the aromatic amino acids, and of course we have some of our nonpolar amino acids in here. But things like glycine and alanine are actually marked as preferring our more polar environments, right? So just because the side group is nonpolar doesn't mean that the amino acid overall is nonpolar. And of course, our charged amino acids are going to have the highest preference for that polar solvent, because there we'll have the water molecules interacting with our charges and helping to stabilize those charges there through hydrogen bonding and interacting with our partial negative and positive charges of our water, as we saw in our previous lecture. Now, of course, as I mentioned earlier, another key function of our amino acids is the ability to react in acid-base reactions, right? So each amino acid has a carboxylic acid, which is relatively acidic, so it can react and donate our protons, and then, of course, become negatively charged. So we refer to this as an ionization of our amino acid. We also have our amino group that is basic. So our amino group will tend to accept protons and become positively charged. So these will have a key effect on our amino acids and our proteins, as we'll be able to donate and accept protons from our amino acids. We'll take a closer look at the ionization of our amino acids and these acid-base reactions in our next video.